The more researchers know about how the coronavirus attaches, invades and hijacks human cells, the more effective the search for drugs to fight it. That was the idea to be true when we began building a map of the coronavirus 2 months ago. The map shows all of the coronavirus proteins and all of the proteins found in the human body that those viral proteins could interact with. In theory, any intersection on the map between viral and human proteins is a place where drugs could fight the coronavirus. But instead of trying to develop new drugs to work on these points of interaction, we turned to the more than 2,000 unique drugs already approved by the FDA for human use. We believed that somewhere on this long list would be a few drugs or compounds that interact with the very same human proteins as the coronavirus. We were right. Multidisciplinary team of researchers at the University of California, San Francisco, called the QCRG, identified 69 existing drugs and compounds with potential to treat COVID-19. A month ago, we began shipping boxes of these drugs off to Institut Pasteur in Paris and Mount Sinai in New York to see if they do in fact fight the coronavirus. In the last 4 weeks, we have tested 47 of these drugs and compounds in the lab against live coronavirus. I'm happy to report we've identified some strong treatment leads and identified 2 separate mechanisms for how these drugs affect SARS-CoV-2 infection. Findings were published on April 30 in the journal Nature. The testing process, the map we developed and the FDA drug catalog we screened it against showed that there were potential interactions between the virus, human cells and existing drugs or compounds. But we didn't know whether the drugs we identified would make a person more resistant to the virus, more susceptible or do anything at all. To find those answers we needed 3 things: the drugs, live virus and cells in which to test them. It would be optimal to test the drugs in infected human cells. However, scientists don't yet know which human cells work best for studying the coronavirus in the laboratory. Instead we used African green monkey cells, which are frequently used in place of human cells to test antiviral drugs. They can be readily infected with the coronavirus and respond to drugs very closely to the way human cells do. After infecting these monkey cells with live virus, our partners in Paris and New York added the drugs we identified to half and kept the other half as controls. They then measured the amount of virus in the samples and the number of cells that were alive. If the samples with drugs had a lower virus count and more cells alive compared to the control, that would suggest the drugs disrupt viral replication. The teams were also looking to see how toxic the drugs were to the cells. After sorting through the results of hundreds of experiments using 47 of the predicted drugs, it seems our interaction predictions were correct. Some of the drugs do in fact work to fight the coronavirus, while others make cells more susceptible to infection. It is incredibly important to remember that these are preliminary findings and have not been tested in people. No one should go out and buy these drugs. But the results are interesting for 2 reasons. Not only did we find individual drugs that look promising to fight the coronavirus or may make people more susceptible to it, we know, at a cellular level, why this is happening. We identified 2 groups of drugs that affect the virus and they do it 2 different ways, one of which has never been described. Disrupting translation at a basic level, viruses spread by entering a cell, hijacking some of the cell's machinery and using it to make more copies of the virus. These new viruses then go on to infect other cells. One step of this process involves the cell making new viral proteins out of viral RNA. This is called translation. When going through the map, we noticed that several viral proteins interacted with human proteins involved in translation and a number of drugs interacted with these proteins. After testing them, we found 2 compounds that disrupt the translation of the virus. The 2 compounds are called tRNATIN4 and zodatifin. Both of these are currently used to treat multiple myeloma and seem to fight COVID-19 by binding to and inhibiting proteins in the cell that are needed for translation. Plitidepsin is a similar molecule to tRNATIN4 and is currently undergoing a clinical trial to treat COVID-19. The second drug, zodatifin, hits a different protein involved in translation. We are working with the CEO of the company that produces it to get it into clinical trials as soon as possible. Sigma receptors, the second group of drugs we identified, work in an entirely different way. Cell receptors are found both inside of and on the surface of all cells. They act like specialized switches. When a specific molecule binds to a specific receptor, this tells a cell to do a specific task. Viruses often use receptors to infect cells. Our original map identified two promising MV cell receptors for drug treatments, Sigmar-1 and Sigmar-2. Testing confirmed our suspicions. We identified 7 drugs or molecules that interact with these receptors. 2 antipsychotics, haloperidol and melphalan, which are used to treat schizophrenia, showed antiviral activity against SARS-CoV-2. 2 potent antihistamines, clemastine and cloparistine, also displayed antiviral activity, as did the compound PB28 and the female hormone progesterone. Remember, all these interactions have so far only been observed in monkey cells in petri dishes. At this time we do not know exactly how the viral proteins manipulate the Sigmar-1 and Sigmar-2 receptors. We think the virus uses these receptors to help make copies of itself, so decreasing their activity likely inhibits replication and reduces infection. Interestingly, a 7th compound, an ingredient commonly found in cough suppressants, called dextromethorphan, does the opposite. Its presence helps the virus. When our partners tested infected cells with this compound, the virus was able to replicate more easily, and more cells died. This is potentially a very important finding, but more tests are needed to determine if cough syrup with this ingredient should be avoided by someone who has COVID-19. All these findings, while exciting, need to undergo clinical trials before the FDA or anyone else should conclude whether to take or stop taking any of these drugs in response to COVID-19. Neither people nor policymakers nor media outlets should panic and jump to conclusions. Another interesting thing to note is that hydroxychloroquine, the controversial drug that has shown mixed results in treating COVID-19, also binds to the Sigmar-1 and Sigmar-2 receptors. But based on our experiments in both labs, we do not think hydroxychloroquine binds to them efficiently. Researchers have long known that hydroxychloroquine easily binds to receptors in the heart and can cause damage. Because of these differences in binding tendencies, we don't think hydroxychloroquine is a reliable treatment. Ongoing clinical trials should soon clarify these unknowns. Treatment sooner rather than later. Our idea was that by better understanding how the coronavirus and human bodies interact, we could find treatments among the thousands of drugs and compounds that already exist. Our idea worked. We not only found multiple drugs that might fight SARS-CoV-2, we learned how and why. But that is not the only thing to be excited about. These same proteins that SARS-CoV-2 uses to infect and replicate in human cells and that are targeted by these drugs are also hijacked by related coronaviruses SARS-1 and MERS. So if any of these drugs do work, they will likely be effective against COVID-22, COVID-24 or any future iterations of COVID that may emerge. Are these promising leads going to have any effect? The next step is to test these drugs in human trials. We have already started this process, and through these trials researchers will examine important factors such as dosage, toxicity and potential beneficial or harmful interactions within the context of COVID-19. Thank you for following Inspire the Future channel. Remember, you can give us the greatest support by subscribing to our channel, liking our videos and also clicking the links on the description side of this video.